Okay, so we continue with these light-independent reactions, which is also known as Kelvin cycle. Okay, now some books use the term dark reaction, which is not a correct term because it gives us the idea saying that this reaction actually happened during the at the night times, which is not correct. In fact, light-dependent reaction take place during the daytime. It just that this process, they are not require the light energy directly. So that's why we say that light independent reaction, whether the light is there or not, this process, they depends on the ATP as well as reduced NADP to function. Okay, so if you compare the Kelvin cycle to the Krebs cycle, quite similar because the starting material at the end of the process regenerate back. Okay, we regenerate back the original starting material. Okay, and we have the molecule that enter into the cycle and exit the cycle. Okay, so Krebs cycle is a catabolic, meaning you break down the substances, correct? Okay? Oxidizing the acetyl-CoA, or more precisely, actually, we oxidize the acetyl group. Change it, guys. Change to acetyl group rather than acetyl-CoA. Okay, they oxidize the acetyl group, and ATP is synthesized. But in contrast, Kelvin cycle is anabolic, means that we want to produce organic carbon compound, and ATP is required in this process. Okay, so what is the functions of light independent reaction of Kelvin cycle? The function actually is to convert carbon dioxide into organic carbon compound. So carbon dioxide is inorganic. So basically, plants want to convert this inorganic carbon compound into the organic carbon compound. So this reaction occurs in stroma, the fluid field area of the chloroplast. But I prefer you guys to write in full stroma of chloroplast. Okay, when the question asks you where is this reaction take place, you don't write stroma only, but you write you write stroma of chloroplast. Okay, and this reaction highlights we need to have ATP and reduce NADP. We have no choice. They must have these two, and these two come from light-dependent reactions. And this Kelvin cycle contains the four main stage. The first three actually within the cycle. Okay, this cycle within this cycle. Okay, so we have the first one, carbon dioxide fixation. Second, we have reduction phase. Third, we have regeneration of carbon dioxide acceptor. And last, we have product synthesis. Now I use different color because product synthesis take place out of the cycle. It's not really inside the cycle. Are you clear? Okay, so I'm going to draw this Kelvin cycle for you guys, but you guys don't draw anything yet because at the end I will do the modifications. So don't draw anything. So wait until I have done. Okay, Okay, so let us look at this Kelvin cycle. What we need to know for Kelvin cycle. So first, for Kelvin cycle, we need to have this carbon dioxide acceptor. Carbon dioxide acceptor means that they accept carbon dioxide because we want to convert CO2, carbon dioxide, into organic carbon compound, such as sucrose, sugar, okay, starch, and so on. Okay, so first of all, we have this molecule called ribulose bisphosphate. Okay, or commonly known as RUBP. So this RUBP is 5 carbon compound. RUBP, 5 carbon compound. So this RUBP will accept CO2. So CO2 in this case, 1 carbon. Can I see that? So the enzyme that involved in these reactions is known as Rubisco. Rubisco stands for ribulose bisphosphate carboxylase. Are you clear? Rubisco, it's a short form. Okay, so it's a ribulose bisphosphate carboxylase. So means we add the carbon to this ribulose bisphosphate, okay, RUBP. So after this reaction take place, they form this what we call the unstable 6-carbon compound. The intermediate compound, 6-carbon intermediate compound. And this 6-carbon intermediate compound immediately split, okay, into 2 molecules of glycerate 3-phosphate. Or commonly known as GP, which is 3-carbon. Are you clear? So it means that if I put in the number of molecules, so 1 molecule, don't write anything yet, 1 molecule of ribulose bisphosphate will react with 1 molecule of CO2, you get 1 molecule unstable 6-carbon compound. So this 6-carbon compound now splits into 2 molecules of the glycerate 3-phosphate. Clear until this part? Okay, so this part, okay, the first process have done. Okay, first process have done. So I label first process, step 1. So what is the step 1? Let me write down. So process number 1 or step 1. So step 1 is known as the carbon dioxide fixation. The idea of fixation basically means that we can't directly, a plant can't directly use CO2. They have to take in the CO2. Okay, they cannot straight away CO2 convert to carbon compound. They have to go through a series of processes. So the first process is termed CO2 fixation. So what are the main events here? So main events are RUBP together with CO2 with the presence of Rubisco. So you get 6-carbon unstable compound. So this 6-carbon unstable compound split into the 2 molecules of GP, which is 3-carbon. Okay, so basically this is step 1, CO2 fixation. Okay, now we look at step 2. Step 2 is we call it reduction phase, where glycerate 3-phosphate is reduced to triose phosphate. Reduce to triose phosphate. Triose phosphate we have learned is 3-carbon, so it means that it's still 2 molecules. Okay, 2 molecules of glycerate 3-phosphate will be reduced. So in this case, who is the reducing agents here? Who reduce it? So in this case, guys, we need to have the reducing agent. So the reducing agents here is reduce NADP. Okay, reduce NADP, so be return back, regenerate back NADP. How many molecules? So 2 molecules, so they need 2. And then in this process also require ATP. Okay, require ATP. So 2 molecules of ATP. Now left with 2 molecules of ADP. Okay, so this process involves the reduced NADP, reduced NADP, and also ATP. So this is step 2. So what actually happened during step 2? Okay, I put it all here. Okay, step 2. Step 2 is known as the reduction phase. So what is the main event? The main event is GP, 3 carbon, reduce. So a reduction take place. So GP become TP, 3 carbon. So with the presence of what? With the presence of reduced NADP and also ATP. Okay? So reduction phase actually take place where GP is reduced to TP, and this TP, okay, sorry, this process require reduce NADP and ATP. So basically, 2 molecule of GP, you get 2 molecule of TP. Okay, step 2 done. Now, if you look at this step 2, after step 2, triphosphate, because it's a cycle, right? So to complete a cycle now, sorry, nice. So type A, this is step 3. So in this process, in step 3, what happens here is step 3 is the regenerations of the carbon dioxide acceptor. And this process require, okay, the input of ATP again. Okay, so total we need 6 ATP. So 6 ATP. Require ATP. So become ATP. So with the present ATP, now triphosphate is regenerate back. Are you clear? So it's called regenerations of ribulose bisphosphate. So this is step 3. So step 3 is known as regeneration of CO2 acceptor, which is RUBP. So in this case, what are the main events? So TP now regenerate back, become RUBP. So with the presence of ATP. Are you clear? Clear? Now, how about product synthesis phase? Product synthesis phase is not part of the cycle. So triphosphate actually can undergo product synthesis phase. Okay, product synthesis phase. So this is step 4. So means that 2 molecules of triphosphates actually can combine together because 3 carbon form glucose, for example. So I put it in general, organic carbon compound. So this organic carbon compound actually is glucose, C6H12O6. So it means that 6 carbon, guys, 6 carbon. So in this case, you get 1 molecule. Are you clear? Okay, so I zoom it, make it smaller, then you look at this process. Now we do have a bit of the question here. What is a bit of question? So first of all, let us look at this. If I only have 2 molecules of triphosphate, can you see that? If we only have 2 molecules of triphosphate, so how to regenerate back a ribulose bisphosphate? How I can generate the glucose, which is impossible. So in fact, to generate 1 glucose molecule, we need to have 6 rounds of Kelvin cycle. Okay, in order, okay. Right, guys. So understand this. So to generate, to produce 1 glucose, then we need to have 6 rounds of Kelvin cycle. So means that everything have to times 6. Why? Because you look at this part here, I highlight C6. So this 6 carbon must come from the CO2. Clear or not? So means that we need to have 6 molecules of CO2. So everything now I have to times 6 in order to produce one glucose molecule. So here, oops, I think guys, the layout. Okay, so now I can remove. So I remove all this. Remove everything times 6 now. Okay, so now look at it, at times 6. So it means that 6 molecules of ribulose bisphosphate react with 6 molecules of CO2 to get 6 molecules of unstable 6-carbon compound. And then these 6 molecules of unstable compound now get us 12 molecule of glycerate 3-phosphate. In the reduction phase, you have 12. Okay, now look at this. Out of 12, 2 molecules, out of this 12, 2 molecules will be used to form organic carbon compound. Are you clear? And then the remaining 10 molecules will be used to regenerate back RUBP. And you look at these guys, okay, 10 molecule. Each molecule got 3 carbon, so total we have 30 carbon. Can you see that? So 30 carbon, you divide it by 6, you will get back 6 molecule of ribulose bisphosphate. Clear or not? If you 30 divided by 6, so you get back 5 carbon compound ribulose bisphosphate. And this process again require 6 molecule ATP. Can you see that? So this is a complete process of Kelvin cycle. Okay, so you can take a photo. So recording. So in exam, what we write, so we follow phase by phase, phase by phase. So first in carbon dioxide, okay, fixation. So first, you guys do not need to worry how many molecules, only few of them you need to know. So we say 1 molecule of RUBP react with 1 molecule of CO2 in the presence of the Rubisco, form 1 molecule of unstable 6-carbon intermediate compound. Are you clear? So you write, RUBP react with CO2 or accept the CO2 with the presence of Rubisco, form 1 molecule unstable 6-carbon compound. So Rubisco is 5 carbon, CO2 1 carbon. Okay, so this is what you write. Second, then you say that this 1 molecule of unstable intermediate or 6-carbon intermediate compound split, okay, into 2 molecule of, this one is very important, 2 molecules of glycerate 3-phosphate. Now be careful, glycerate 3-phosphate, you have to spell it out first because the short form we use is GP. Okay, so therefore you need to actually write it down. Okay, and this is very, very important to show you guys know. So number actually very important here to show that you understand. Okay, this unstable carbon compound is 6 carbon split into 2 molecules of the glycerate 3-phosphate. So this thing happened during a CO2 fixation. Okay, now next, after CO2 fixation is a reduction phase. So reduction phase Very simple, what you write. So we just say that one molecule of GP is reduced to one molecule. You want to put the 3 carbon, it's up to you. So one molecule of GP is reduced to one molecule of triose phosphate. First time I will write full triose phosphate. So TP. So 3 carbon. Okay. With the presence of reduced NADP and ATP. Don't need to mention how many molecules. Are you clear? One molecule of GP is reduced to one molecule of triose phosphate with the presence of reduced NADP and ATP. This is the reduction phase. Okay. Next, we have the regenerations of CO2 acceptor, which is RUBP. So again, with the presence of ATP, this one ATP only. So TP is ATP are converted to RUBP. Just write like this, enough already. Do not need to mention how many molecules. Okay, if you want, okay, totally up to you. Okay, so this regenerations of the CO2 acceptor. Okay, and then product synthesis phase. So we just need just need to write TP. Okay, is converted into sugar or carbohydrates. You can say carbohydrates. So such as the glucose, starch, lipids. Okay, so it can be the glycerol and fatty acid. Okay, and also amino acid. Are you clear? So not the amino acid and proteins. Okay. So this is product synthesis phase. Okay. So zoom in, then you can have the full process. Okay, so I want to highlight the compounds that we guys need to know. First, ribulose bisphosphate. Can you see that? RUBP, glycerate 3-phosphate, GP, triose phosphate, TP. These 3. Okay. Now, in order to get the complete cycle, so I prefer to change this. Remove. Sorry, guys. So number here, I can put it in now. It's actually 6 molecule. 6 molecule. This one is 6 molecules, short life intermediate. And then your 12 molecule of glycerate 3-phosphate, your 12 molecules of triose phosphate. Then we take 2. Okay. And then we take 10. Okay. to form back the ribulose bisphosphate. So these two outputs will form glucose. Okay, so what you need to do now, complete this paragraph. Okay. Okay, Kelvin cycle incorporate carbon dioxide and into this ribulose bisphosphate. So the enzyme that involved in this step is called ribulose bisphosphate carboxylase, or in short, we call it Rubisco. It is the most abundant protein in the chloroplast and also thought to be the most abundant proteins on the earth. So the product of this reaction is a 6-carbon intermediate, which is unstable because it's so energetically unstable that it immediately split it to half, forming 2 molecules of glycerate 3-phosphate for each CO2 fix. So next, reduction phase. Each molecule of GP receive additional phosphate group from ATP, become glycerate 1,3-bisphosphate, but you do not need to know about this. Then it's reduced by the reduced NADP, becoming glyceraldehyde 3-phosphate, which is also known as the triphosphate. So be careful about this term, guys. Sometimes you look at a certain book, they use the word glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate actually refer to triphosphate. Be careful, this is glycerate. Okay, in some book they use the word glyceraldehyde 3-phosphate, so which is mean triphosphate in our syllabus. So TP is a sugar, the same 3-carbon compound formed in the glycolysis by the splitting of the glucose. For every 6, can you see that, molecule of CO2 that enter the cycle, there are 12 molecules of TP formed, but only 1 molecule of TP can be counted as net gains of the carbohydrate because the rest are required to complete cycle. Sorry, guys, my mistake is with 2. Change it here, 2. Okay. Only 2 molecules of TP can be counted as a net gains. Yeah. So the cycle with 30 carbons worth of the carbohydrate in the form of the 6 molecule of 5-carbon sugar RUBP. Now there are 36 carbons worth of the carbohydrates because 30 RUBP, 6 from the CO2. So total we have 36 carbon. Okay, so 2 molecules exit the cycle to be used by the plant cells, but the other 10 molecule must be recycled to regenerate the 6 molecule RUBP. So in a complex series of reaction, the carbon skeleton of 10 molecule TP will rearrange by the last steps of the Kelvin cycle into 6 molecule RUBP. And this process again require ATP, 6 molecule. So for the next synthesis of 2 molecules, 2 TP molecules, the Kelvin cycle consumes a total 18 molecules of ATP and 12 molecule reduced NADP. So the light-dependent reaction generate ATP and reduce NADP. So the TP spun off from the Kelvin cycle become the starting material for the metabolic pathway that synthesizes other organic carbon compound, including glucose and disaccharide, sucrose, and other carbohydrates. Okay. So the reactions of Kelvin cycle provide a net yield of 3 carbons phosphorylated sugar, which is TP. So TP can convert into the glucose and also the starch. Some TP can be converted to amino acid and fatty acid. Okay, some TP can be exported to the cytosol where enzyme convert it into sucrose and translocate from the leaf all the way to the root. Okay, so then at the roots, for example, sucrose can be broken down for aerobic respiration or become the starting compound to make the various organic molecule. So what are the important carbohydrates in plants? For example, we know cellulose, we have the starch, okay, that is particularly important in the roots and also the tuber, such as the potato tuber. Okay, so this diagram, just for your information, you can see that we have the carbon dioxide fixation in the Kelvin cycle where CO2 coming in. And then you can see that it generates the 6-carbon sugar phosphate, and you do have the triphosphate, pyruvate, and so on. So this they call as the metro, okay? Metabolic metro map is something like a stations, the RT stations or the MRT station. So you can see that you have a urea cycle, a lot of chemical reaction actually take place here. Okay, but for us, we do need to know here only with the light, okay, produce the ATP and reduce NADP, channel them to the Kelvin cycle where CO2 enter and continue the Kelvin cycle. So the product of Kelvin cycle you have the hexose P, hexose phosphate. Hexose phosphate is the glucose phosphate, triose phosphate. So triose phosphate go all the way oxidized. You can see that you get a pyruvate, and then acetyl-CoA, then into the Krebs cycle. Acetyl-CoA go into the Krebs cycle. So where you can have the ATP synthesis take place. Okay, so again, this is just for information only. If you're interested to know more, you can search it. Okay, so that's all for today class. I'm going to stop the recording now.